Vinita. In our industry, at Aarti Industries, we are discouraged to say sir, uh, but they have left us to it's individual's choice to say sir. But there is a strong culture of discouraging to call anybody sir. It's directly name is good to call. So I will also discourage all students here. Do not call me sir. You can call directly me Vinod. That is very clean. Uh, Going forward, what I was discussing before the start of this session was this can be a very interactive session. No need of one way. I come and talk, and you guys listen wherever you feel like you would like to understand more deeper aspects of what is going on in this presentation. You can anytime stop me and ask the questions, and let this be interactive session. I am presenting my screen. Hope you guys are able to see my screen.
Yes.
Yes, sir. Yes.
Yeah. Thank you very much for the confirmation. So the topic I have chosen is process development and technology readiness level. We were discussing the reason why I chose this particular topic of process innovation is it is very important aspect in terms of Indian pharmaceutical industry growing in pharmaceutical area. uh, based on the generic the compounds that are going generic. Immediately pharmaceutical industry is very proactive in innovating the processes, turnaround of the processes, by of bypassing the infringement, and preparing these molecules. The more important aspect is preparing these molecules in very economic way, so that these molecules could be sold in any competitive market at lower cost. That's the advantage, market advantage, process innovation advantage that we have here. And the example which I have picked up is roafovir etalafenamide, a very complicated name of pharmaceutical products I have seen, but this is also one of them. So for the sake of simplicity, what I will call it as an amide, for the sake of understanding and free communication. What you will find in this presentation is understanding about various levels of technology readiness, roafovir etalafinamide process development from TRL level 1 to TRL 4, and what were the challenges that faced after fourth level, and how the development happened in various steps of synthesis, and then specific challenges that were faced in terms of impurity, achieving purity, decreasing impurity, and what was the result state after passing a level of 7. And then we'll see how engineering activities also play a very critical role in process innovation. To start with, uh, these are the general classification scales: lab scale, pilot scale, and manufacturing scale. You can term this technology readiness level from 1 to 4, club them together into a lab scale, wherein the process basic principle is observed, it is then conceptualized, proof of concept is seen, and technology validation is done in the lab. It's not so easy as I am speaking here. It takes years for the project to start come from TL level 1 to TL level 4. After crossing this path, it enters into a pilot scaling from TRL level 5 to TRL level 7, wherein the technology is validated. Here the change is, earlier the technology was validated in laboratory. This time it is validated in relevant environment. Environment in terms of, say, it is just a kind of seeing at a larger scale as compared to the lab scale, and therefore the relevant environment could be carrying out the reaction or the process innovation in bigger scale. Say for example, it may vary from kg to few tons. It depends on the organization's capacity at which they would like to perform and understand the process aspects of the preparation. Then there enters the validation, which was done in TRL level 5, is then demonstrated in the relevant environment. There is back and forth happening from TRL 5 to TRL 6. Learnings of validations are incorporated, and then once the learnings are all incorporated and there are certain understandings about the process, and there could be reduced number of challenges as compared to TRL level 4 when it started up in after laboratory process. The challenges and the issues could be more resolved here. And then once the prototype and a prototype is demonstrated in operational environment, it goes to the manufacturing trials where systems, infrastructure requirements, all those things are qualified, and actual system is proven with the help of manufacturing trials, and there could be few corrections. There could be asking data from R&D. What if study could also be entering at this point of time, and there could be certain manufacturing trials which may require certain understandings, process understandings. What happens if there is particular change in certain conditions? Holding time is increased. So all those studies are done at manufacturing trials, and after that there is a commissioning of manufacturing. Before the commissioning of manufacturing, there is obviously obvious way of asserting the safety parameters, wherein the entire plant is checked upon for various safety parameters in terms of infrastructure requirement, in terms of hazardous understanding, in terms of special care requiring to avoid runaway of the reactions, hotspots within the reactions. You may think of exothermicity or endothermicity leading to a pressurization of reactors. All those aspects of the process innovations are then considered at this point of time if they were missed out in the piloting stage. Any questions here? Anybody is having questions here, or?
I think there are no questions yet.
Okay.
But we'll request the participants so they can post their questions in chat box. So
Okay.
we'll take them in.
You can.
Yes.
Will you please read those questions if they are posting in the chat? I would be
Sure.
able to see those questions. You can just stop me and read out those to me. We can address those questions, because then it will be very useful for them to understand the concept clear here, and then we can move ahead.
Right. Yes.
So this is the synthesis of the molecule amide, which we saw, which I narrated in the introductory phase. The synthesis is in a resolved state because the product was already developed. It was identified to be a lead molecule in terms of activity against antiretroviral activity against immunoviruses. Uh, the process that was optimized after the scale, after TLR1 level and to TRL4 technology validations, the process was the compound 3 was converted to compound 4 by decarboxylation, and the phosphine was introduced using triphenyl phosphine. THF was used as a solvent. I am highlighting specific requirements of this reaction so that when we go further, these highlights, you can recollect these highlights, and then we can connect to those subsequent slides. Re-slurry in MTB was required, the solvent volume, because when it comes to the scalability, volumes of solvents need to be really low. For example, if you have to produce, the throughput of your product has to be high, then the solvent quantity needs to be really optimized. But in this laboratory scale, re-slurry in MBT, methyl butyl ethylene tertiary ether, this particular solvent was used at a ratio of 135 liters per kg. Reslurring of so that reduced the throughput when it comes to the uh, scaling up of the process. And when this compound 4 was prepared, it was reacted. The reaction is called as iodo trans etherification reaction, wherein iodine has to be used and there were several challenges even during the piloting and even during manufacturing. This was advanced stage piloting, I would say, not manufacturing. This iodine was inevitable. Use of iodine was inevitable, and therefore, despite the challenges in terms of its approvals and in terms of its use, this iodine has to be used here. Basically, what happens because of the use of iodine is it triggers the polymerization of THF, and the polymerization of THF then polymerized product, when the workup is conducted of this reaction, this polymerized product causes a problem in the filtration, wherein the filtration time taken would be somewhere around 24 hours. Further, it involved the use of molecular sieves to reduce the water content within the reaction, because the compound number 6 that you can see here can easily dehydrolyze to corresponding phosphates, and for that reason molecular sieves need to be used. But the problem with the use of molecular sieves was when the reactions were performed, they pulverized easily to form a powder, again causing a problem of filtration and workup during the reaction. And then 6 was converted to compound 7, which was amorphous. Amorphous precipitation led to a problem that it cannot be further purified, and it had many impurities, because of which the compound number 1, which is prepared from compound 7, is not pure, and it contains various other impurities, and which need to be uh, removed by way of silica gel technology, gel filtration, silica gel chromatography, which obviously is not a scalable technology in terms of plants. And then this compound number 1, which is our interest molecule, having impurities, wherein the fluoro, which you can see at the second position, is removed or is knocked off during the course of the reaction, to give us a citrate, which was reacted with citrate. The important aspect here is, again, for the purification there has to be a re-slurry in acetonitrile twice, so that the compound could be purified. So there were several challenges. Despite that, the lab validations and all those activities were done, there were several challenges at the scale of technology readiness level 1 to technology readiness level 4, which were unresolved. And the compound was prepared, though the multi-kilogram quantities of API was prepared for the sake of testing at various levels in clinical phases, clinical 1 and 2. By the way, I just would like to mention this molecule is currently under phase 2 development for anti-retroviral drugs, and and it is being tested as a monotherapy for the therapies where the existing therapies are failing, and the resistant cases, HIV resistant cases are being they are checking whether this could be useful in addressing those resistant cases. The compound was prepared and tested in laboratory scale in this manner. Though not a scalable process, this was very useful for them to carry out clinical phases. Now the challenges that were left, as I discussed earlier, scalability was the problem because the huge quantities of solvents had to be used. Then, because which reduced the operational capacity of plants, and process to mass intensity was low. The various products obtained, intermediates obtained, like compound number 7, they were amorphous, and the achieving the desired purity was a problem. The compound formed by defluorination, desfluororoafiravir, which you can see here in this slide 8, where the F is knocked off, fluorine is knocked off, is at 2000 ppm, where the acceptable permissible limit was later on identified at 100 ppm. So it's for that removal of that desfluoro, there was heavy reliance on silica gel chromatography to attain the purity, but still that failed, and there were the challenges unresolved. And for that, the further development of this process had to be taken up to enter into the subsequent phase of piloting and detailed studying, wherein we'll see how the technology validation in relevant environment was done. Technology then demonstrated and prototypes were prepared to address all these challenges. Now coming back to the first step, we'll narrow down, zoom in down with the detailed aspects of the challenges that we were facing when it comes to preparation of compound number 4. In these four, as I told earlier, THF solvent was used. The solubility of compound number 3 was lesser in THF. That was one problem. Second problem was fluorine, compound number 4 used to recrystallize easily. That high crystallization was observed in THF solvent, and because of this, the reaction time was around 12 hours, slow reaction rate basically. And then MTBE solvent was used for isolation of 4, and then for purification, high volume of over 135 liters per kg were required to remove the byproducts that were from use of phenyl triphenyl phosphine and DIAD. Further, the ratios were higher, 3.2 to 3.4, and the yield losses were also higher because of the high use of MTBE solvents. When the R&D was working on this is how the approach and the development of your technology readiness happens. You have to break down your problems to zero down your problems to each one of step. Each problem has to be identified and the solution has to be provided, which is exactly done here. To improve the technology readiness level from which was earlier at TRL level 5, it has to be taken to TRL level 7. Then each and every problem has to be addressed in a subsequent way, which you can see here also. The extensive solvent screening was done, and acetonitrile was found to be a more suitable solvent because it was compound number 3 was easily soluble in the reaction mass, and also there wasn't rapid crystallization of compound number 4, and the result as a result there was enhanced rate of reaction, and the reaction time reduced from 12 to 2 hours. The first effect of acetonitrile. The second thing what the changes and process innovation did was seeding this compound number 4 in after the completion of the reaction, then changing the solvent from acetonitrile to isopropyl acetate. I'm just naming the solvents, but identifying these solvents in terms of when you are actually working and improving the processes is a real challenge for any chemist or any person from who is working or associated with R&D. Isopropyl acetate, identifying isopropyl acetate as a solvent exchange, and then using anti-solvent and heptane was the technology that they used, because of which the re-slurry volume of MTB was reduced from 135 to 30 liters per kg. Huge improvement in scalability can be seen here. Then the reduction of triphenylphosphine and DIAD from 3.2 to 3.4, 3.2 to 2.5, and 3.4 to 2.7 respectively, only added up to the lesser cost, because the cost could be reduced. That is one point. And also impurity profile or removal of these byproducts also was easy, and the all things started improving in terms of handling of these byproducts. And overall cumulative effect of this improvement resulted into the 70 to 80% yield of the product, which was heavy losses were seen as earlier, and the purity was also higher to 90 to 95 percentage. Hence there was substantial improvement in step 1 and technology readiness of step 1 from 4 to taking up to the level of 7. Now coming to technology readiness of step 2, there were several problems identified. I have highlighted few of them, like once the THF used, this has to be added. The compound number 4 has to be added slowly. That was first problem, because slow addition of compound number 4, because of its lesser solubility, it has to be added in excess quantity of iodine. Molecular sieves and THF had to be used. The problem with this mixture is there are occupational hazards associated with the use of iodine and THF polymerization levels. Also, the cryogenic condition of -10 to -12 had to be utilized. Molecular sieves, as I told earlier, they pulverized, which were used for absorbing or preventing decomposition or degradation or dehydrolysis uh, of this compound 6. Um, Iodine initiates polymerization, which again leads to the filtration issues. Compound 6 isolated was amorphous. Because of amorphous, requisite purity could not be achieved. Now the simple change from the use of THF as a solvent to a dichloromethane as a solvent firstly avoided the incompatibility issue of iodine and THF, thereby improving the TRL level of uh, from 5 to 7, which ultimately resulted into the enhancement of percentage. If and the scientists also learned that if the mode of addition or the rate of addition of compound number 4 to 5 and iodine and THF is kept at 10, kept between 10 hours, the yield achieved is as high as 78 percentage. Because of the use of dichloromethane, cryogenic conditions -10 degrees Celsius were no longer required, and the reactions could be performed easily at room temperatures, thereby avoiding the operational expenditures. In short, we call OPEX. And this problem of amorphous compound number 6 was uh, changed to the succinates, and all those could be prepared, and the crystalline structure was easily achieved there, thereby getting the purity after MEK isolation to the purity of 95%. So step by step, steadily addressing step 1 and step 2, the prospects of this process for scalability were improving. But this wasn't enough. And there were several challenges lying ahead in terms of step 3, in step 4 and 5. There were not much challenges here, but the very important changes what happened in this process was was the reduction in the overall volume of oxone to 25 liters per hour, because of the mode of additions. And telescoping is also very important aspect when it comes to process innovation. Let me explain what this telescoping is. For example, there is a compound which you are preparing in multi stages, A to B to C, and then you go to D. In many processes or batch processes, intermediates B and C need to be isolated, purified, and then again added to another reactor, and this is how the compound C formed will also be isolated, purified, and then used for the preparation of compound D. But when it comes to telescoping, compound B and C are not isolated. They are in the same pot with same solvent systems or reagent systems. They continue to be present in the reaction without impacting the overall performance of the product in terms of its purity, in terms of its rate of reaction or scalability. So all these parameters put together, telescoping is very important because it saves on manpower, it saves on operational expenditures. So this was very important for improving the technology readiness of this process. Uh, Technology readiness of this process. Step number 3 to the next level. Now that there was substantial improvement in first 3 steps, there were challenges. The earlier route utilized acetic acid, and then silica gel chromatography had to be used, and citric acid was also used to prepare the citrate of amide. The problem associated with the use of acetic acid was there was partial decomposition of the product, resulting into the phosphate corresponding phosphate formation. Also, extra care had to be taken when it comes to avoid the decomposition during the workup. So no extra heating was to be taken care. Then the product drying was done using magnesium sulfate. Compound number one was purified using magnesium sulfate, uh, which again magnesium sulfate, though not a big challenge here, but they identified this challenge as a result, maybe because this wasn't sufficient enough uh, to address the problem of decomposition of the product, and therefore there has to be a better uh, uh, water absorbing reagent. That was also the challenge that was underlying, and the product was subjected to silica gel chromatography to remove compound number 8. The yield obtained here was only 40%, and 2 slurries required for crystallization. So amorphous compound getting into crystallization, heavy losses of yields, uh, uh, decomposition, formation of impurities because of insufficient or inefficiency of water absorbing capacity of magnesium. Because this compound one is very sensitive towards water. The improvement in terms of technology readiness could be achieved because of the use of ethyl acetate solvent when combined with methyl ethyl ketone. The chemoselectivity of this reaction of 7 to 1b was achieved, and 1b is a vinylate salt of amide, which was then with simple use of ethyl acetate water biphasic system, monophasic system, and potassium bicarbonate resulted into the compound of formation 1. And the telescoping, as we said earlier, also added to the advantage of this step as well, and the yield thereby improved to 65% and the purity to 97%. So now while I was discussing the improvement in technology readiness from lab scale to this level, each and every process here, whatever improvement they had done, is a subject matter of patent application, because the molecule is new, the process they are developing is new. uh, the the process which we saw till TRL level 4 is also patentable, but there could be improvement in processes, and this well developed process can also go for patent application. The point here, despite the use of silica gel chromatography for purification of this mitochondrial toxin impurity, as you know in pharmacology, you study that impurities and impurity profiling plays very critical role for launching of or any API to be considered for human trials and utilization. This product was formed at 2000 ppm as against the requirement below 100 ppm, and for that simulated moving bed chromatography technology was used. This is very common technology and as I know, in chemical segment, Torre has developed this technology for its dichlorotoluenes, and there are many other companies who have developed this technology, but uh, this is not so commonly available technology, and therefore to make it more scalable and more easy, uh, there was a need of improvement of identifying. Now the number of problems, as we have seen earlier, from level 4 to level 7 have now reduced, and there are very specific problems that are coming up, like say impurity, which is very specific and very critical parameter, and those parameters are also addressed to improve the readiness of this process for further handover. uh, Now that SMPC is not suitable because of expertise that is required in terms of skilled manpower, and there was further investigation into several pathways uh, of identifying why this compound number 8 is prepared, and the design of experiment was worked upon. It's not easy, but let me also tell you there are various software who can help you with designing your experiments, and those with the help of these experiments, it was identified that compound number 12, which you can see here, is the is the uh, is the starting material to get to compound 8, which initially gave the control that if the compound 12 formation of compound 12 is controlled, automatically the formation of compound 8 can also be controlled. And how that is to be done with how that is done came up after several analysis, design of experiments, and that was identified as increasing the volume, increasing the ratio of compound number 5, which is the reactant, from zero point 0.6 to zero point 0.7. Minor adjustments in terms of solvent volumes, those are utilized in that particular reaction. Decreasing the temperature to 12 degrees Celsius. Initially, it was carried out at 25 around temperature, and slow addition of compound number 5, uh, say around 5 hours time period. These all cumulated resulted into the lower formation of compound number 12. And then, as initially mentioned, there was also a problem of inefficient absorption capacity of magnesium sulfate uh, for reducing the hydroscopicity of the product or the reaction mass. The reaction mass was then filtered through cellulose depth filter, which improved the absorbing capacity, reduced the water content below 5%, 0.5%, and then because of these changes, there was very minor hydrolytic degradation, which improved the impurity profiling, and which also resolved the problem of bringing this impurity below 100 ppm. So resolved state of pilot scaling, which we can state is impurity, base fluoro impurity, which was mitochondrial toxicity, was reduced to below 100 ppm. Selectivity of desired isomer achieved. Magnesium sulfate for drying avoided, and the better technology in terms of use of cellulose depth filter was utilized, which again contributed to the reduced decomposition of the products or intermediates. Impurity and yields at various stages improved. Reaction rate of reaction improved. As we can see, there were improvement in terms of when the solvent was changed from THF to dichloromethane. Reaction temperature was reduced from 12 to 2. Ratios of solvents like MTBE and other purification solvents were also reduced, which enhanced the throughput. Issues like solubility of reactants because of use of dichloromethane, the solubility of products and all those problems were also issued, which brought homogeneity in the reaction. And the various temperature conditions were optimized. Like we said, 25 degrees Celsius to 12 degrees Celsius reduced the formation of compound number 8. Those all conditions were also optimized. So this is a resolved state wherein we can say that now the technology which was given us at a stage of technology validation at lab scale is now gone various phases of level 5, 6, and 7, and has come up with a level of 7. Now there is an improvement, and parallel activities can also go when it comes to the engineering parameters. Now this taking forward, the engineering team comes in picture. Technology transfer activities also parallelly initiates when we were saying how this readiness from 5 to 7 is coming up. At that point of time, at back end, there are engineers who are also parallelly doing the results of the processes, They are also evaluating the results, and they are also back end preparing their own studies. And they might be further asking about what if studies by technology transfer team to ascertain the suitable way of implementing this technology or scaling up this technology. Basic engineering drawings may also initiate at pilot scale, which now will enter into the detailed engineering phase. Detailed drawings would be ascertained. Process innovation development is now predominated by machinery equipment related challenges, material of construction. What could be the material of construction based on the nature of corrosive nature, or maybe the high pressure reaction conditions. All these parameters are also taken into consideration when the machinery and equipment challenges are addressed. The attention is also given to very minute details, like say for example, what would be the volumes of required in terms of uh, MTB solvents, or like we have seen there are huge solvents or reduced capacity solvents. So what would be the open space or operational capacity of machinery, whether we will be consuming 80% of the overall reactor, or we'll leave 10%, or maybe 70% is to be 30% is to be left as unoccupied during the course of the reaction. That all those detailings are done at this stage. Business case evaluation is done at every stage, and precision is improved from stages to stages. Business case evaluation is basically understanding the potential, because these projects are never for a I would say uh, they are mainly done for a business perspective, and therefore what profits they bring to the organization, what is the value addition, contribution, profit contribution that they will be generating, what are the volumes that will be requiring, what will be the break even point of this process, what are the critical parameters, raw material costing here, and the competitive landscaping. All those things, though have done at earlier stages, they will also be re-evaluated, because by the time the technology readiness comes from 1 to 7 or 9, there is substantial change in the market scenario, and there is substantial change in the competitor level. Competitors might also be evaluating certain other aspects. So from that perspective, or substituted products may also be coming up, which may cost give your product competition in the marketplace. From that perspective, business case evaluation is done at every stages of this development. Then after the business cases are done, there are various levels. I would say this is very extensive task. uh, uh, like there could be class 4, class 3, class 4, class 5 levels of business case evaluations. Then then erection of requisite infrastructure to conduct trials, which obviously starts when the project starts getting into the TRL level 5, readiness level 9. And then when once the erection of the infrastructure is done, requisite all engineering and all these activities are done. Safety assessment is the prime focus, wherein, and also other good manufacturing practices and other requisites are also assessed at this point of time. And there could be trial runs that can happen. At trial runs, there could be another phases, challenges, new challenges that will be coming up because of the increase in volumes and increase in capacities, and wherein mass starts playing a role in terms of behavior. And from that perspective, there could be certain studies that could be coming back to level TRL level 5, or maybe even TRL level 4. And those studies can parallel run. By the time these all studies are completed, and the organization or the company is confident, they can go for commissioning of the manufacturing while addressing the new issues that will be coming after manufacturing. These are all the activities that are usually done. These are more extensive, and maybe these are no way relevant to us, because many engineers are involved here. Chemical engineers play critical role here. It is from pharmacology and maybe from a scientist perspective to provide the data, whatever data they would be asking for. Yeah, that was all from my side.
Yes, thank you, sir. Uh, Actually, we have one question in chat box, so may I take that question?
Yeah, sure.
Uh, so if the impurity which we found at the earlier stage, further we get same impurity in the latter stage, like stage, then at that time, what should we do? Does the whole process we have to repeat from first stage?
So usually there is no such ready-made answer, as we have seen. There could be two approaches of addressing the impurity. One is letting it form and then going for a purification, as we have seen in the level 5, wherein it was allowed to be formed, and then it was tried to be removed with various other technologies like silica gel technology or SMBC technology. But these purities, sometimes we have to always see which approach is suitable to me at that point of reaction or for that particular technology. And if I was able to achieve with SMBC the results with consistent results, the company will not have gone for this detailed understanding of why this is formed. Definitely company goes into that, but then they will not change the other aspects also. So you have to see whether to allow the formation of impurity and they then go for purification, or avoid the formation of impurity, therefore the further stage of purification could be avoided. But sometimes what happens in during the course of the reaction, that impurity which is formed is if not removed in its subsequent stages, it can get removed in say for example, it is not removed in immediate stage, but it can get removed in subsequent workup processes. So sometimes you have to see as a whole picture what happens during the course of the reaction. It's and then study, ascertain, analyze, and see what is very much beneficial for us, and take a call based on the results and analysis.
Okay, thank you, sir. So there is one more question in line of this. So what are the different purification techniques for large scale? So the students mainly know about the lab scale purification methods, but they are not well aware with the manufacturing scale levels.
Yeah, say for example, as we discussed this SMBC, that is also a very good purification method, and that has a capacity to purify very close boiling solvents. So that is very useful technology. But apart from that, like say for example, there could be distillation process, and even if the boiling points are at a difference of say zero point 0.1 to zero point 0.2 levels, increasing height of the column uh, and reflux ratio, increasing the efficiency of your optimizing your bottom temperatures and up temperatures and reflux ratio, magnetic reflexes. Those are also the technologies that can be used. Then resin technology, wherein resinous columns are also used to purify the compounds. Recrystallization techniques are very old techniques that are also used. And then filtration, when it comes to filtration, there could be filtration at various microns, depending on the purity and those aspects. So these are few technologies that are quite greatly scalable. Resiniers are coming up very nicely, and they have been constantly used in the industry. Like say for example, even if the impurities both are in say for example HCl and HF, both are there in gaseous state, then it is a challenge for us to how to get HF separated from HCl. And those, in that case, you have a bed of various resins, or maybe calcium chloride or alumina. You form a bed of those, and you pass that gas through to that bed. Eventually, the gas will be separated, and HF is adsorbed on CaCl2 to convert it into CF2, and HCl is we get purified HCl. So absorption, various absorptions or counter current mechanisms. These are all technologies that are currently being implemented in the industry. Hope this
Right,
answers my question,
right,
your
right.
question.
Yes, sir. So actually in the lab scale, there are different challenges, and in the large scale, there is a different, right?
Yeah.
So like you explain about the purification techniques. So we are also eager to know that how the drying reactions or the anhydrous reaction generally used in the lab, done in the large scale methods.
Yeah. Basically, uh, there are AHF, say for example, anhydrous hydrogen fluoride. Uh, it requires anhydrous conditions. So there are specific solvents like pyridine, which goes gel with maintaining anhydrous conditions. Then you can use nitrogen as a environment within the reactor. A continuous flow of nitrogen can be maintained to avoid uh, oxygen oxygen in the reactor. Then there are certain other solvents that are more compatible to the certain catalyst, wherein they play very critical role in terms of avoiding the contact of that particular catalyst to the oxygen conditions. So those few conditions can be effectively maintained in plant scale in our industry.
Right. Thank you, sir. So now for the students, sir, will you suggest that how any products is first select for the development process? What criteria generally we observed for the before the development of any product? Like for API, you explain all the process, but for any general products, so what we generally see before selecting that product for development?
For any general product, see, the process varies. Detailed nitty-gritties of the process varies from product to product. But if I have to generalize this process for any product, then the steps are like the first step is conducting a literature study, which is very obvious to see what is available in the literature and see where the current technology is existing and identify the need, whether the humanity or the community is in the need of this particular, is there any unfelt need that is left, and can we bridge that gap based on what we have in our hand. So because what i- what what if or capability of my assessments or my assets also needs to be done to check whether I am capable of meeting that unfelt need. So first thing is to identify the unmet need, then to check whether I am in a capacity or in a position to meet that need, and then go at a stages wherein you say how can I meet that need. And from there, there could be the conceptualization of the product at initial stages, and from conceptualization, then the laboratory activities of conceptualization, then in the checking the proof of concept, validation, and then observing the proof of concept of technology development. All those process starts. But basically first thing is to see at the market, see what is required, see I can whether I can meet those requirements, and then go to the technology set. From technology readiness level 1, it starts, the flow is same for most of the processes or products.
Right. And at which stage we should protect our product or process during this whole process of development?
Usually what happens in this pharmaceutical or any industry is when you have started the work and the proof of concept is seen, as we have seen, the proof of concept is seen in technology readiness level 3. That is a stage where usually many companies file the application. Once that is achieved, there could be further improvements, as we saw uh, from the TRL level 4. There were substantial improvement in terms of product impurity and uh, at a stage you can file, say for example, I can think of filing product process innovation at TRL 7 level. But it is my best way to check how late I can file patent application, for the reason that patent applications are having limited life cycle. They are not given for lifetime, and I need to check if I am filing at early stage, I will be losing actually on the milking stage at a product when it is mature. But at the same time, I also have a fear in my mind, if my competitors come up for filing and they take up my invention, there is the analysis that one has to do in terms of what is the market situation, what is the competitor situation, and where do I stand. If you think that you have the reason to believe that you are at an advanced stage as compared to your competitors, then you can choose for filing IP at a later stage. But if you believe that there are close competitors who are operating very closely with my area, then you have to file a patent application first, even before the proof of concept is seen in the laboratory. Many a times what happens is even if I file a patent application fearing competitors, and see that, and later on I came to know that no, there are no competitors that are coming up, then in that cases what we can do is to abandon that particular filing, so that it doesn't comes in public domain, and I get an advantage of another year. That is also the game which Lupin many a times plays. They file the application, they work on the product for a year, and if they find that it is no one is coming up with this technology, they abandon that application and refile the application. The filing, as long as it's not in public domain, it is not seen by anyone. That is the advantage of the IP that one can take off.